use of LNPs is going to become very widespread because of the fact that it has a lot of opportunity and a lot of potential for many different modalities.
Like the vaccine is the biggest example of how advanced the field can be, and like if you look at more rare genetic disorders, which I think most of the companies in this field are working on, it opens an entire spectrum of diseases that you can go forward and treat. So.
Based on existing drug products which are being approved, for example, Onpatro and again Alnylam's siRNA therapeutics for cardiovascular disorders. The LNPs are going to be forefront now, both in gene therapy and small molecule delivery. But mostly brain targeting is one of the new approaches to be addressed using lipid nanoparticles, essentially non-viral vectors, and a single curative therapy that will be best for gene therapy, genetic disorders.
I would think so. I think LNPs are quite powerful. They should be potentially as a mimic of many of therapies that we can put in the patient in various angles. So far, we have seen the impact in vaccination based on the vaccines that unmasked us during this time. And then we do think that in general, they have been researched very well in various areas, from inflammation to cancer biology, cancer immunology, and so on. So there is a whole lot more to do and a lot of these power of LNPs to unleash.
So probably the biggest advantage of LNPs versus other conventional forms of oligonucleotide delivery is it's non-viral, and so you don't have to worry as much about the immune system, and cationic liposomes are going to be more toxic versus LNPs, which are going to be more recognized, especially with recent events.
With AAVs, I think the main concern, as my understanding, is essentially repeat dosing. Namely, once dosed once with an AAV, the concern is that the immune system will recognize it and you won't be able to dose a second time. Whereas LNPs, what we've proven, I think, through the Moderna and Pfizer BioNTech vaccines is that, of course, you can boost, you can dose repeatedly. So
they're very good at delivering RNA to the cytoplasm of the cell where protein is made. They're also degradable, short-lived, and so they avoid some of the potential tolerability issues that cationic lipids have had in the past. And in terms of AAVs versus LNPs, they're redosable, and they avoid some of the potential biological implications for AAV-related responses.
Yeah, so LNP are chemically synthesized, not biologically derived, so that's one advantage, and the other is you can redose them. So regulatory and clinically they are easier to translate than an AAV.
Yeah, I think both the size and PDI are very, very important because it dictates how it is going to enter into your cell membrane and how it's going to help you target different organs within the body. So I think
getting to different tissues, it can often be done with size-dependent characterization. I think the challenge then becomes manufacturability, being able to sterile filter materials so that they're safe for injection. Becomes a little bit more challenging as you're getting away from some of the smaller sizes that people have traditionally used in the LNP space. So yes, I think size can impact distribution, but it comes at a cost potentially depending on how well controlled you can be with that process.
Yes, size is overstated a lot of the time, but anything under, which can be terminally sterile filtered, under 150 nanometers is a viable product. Tissue distribution is more a play of, as we all saw during the conference, charge and composition more than the size.
pegylation is nothing but you are adding another lipid component to it, another layer, so that definitely increases the circulation time. Now if it's going to increase the circulation time, it's also going to increase the amount of payload that could be delivered into the system. But apart from this, it also hampers or compromises the transfection rate. Now for example, that is what I wouldn't say state the ionizable lipid, but a lipid composition, if you're gonna coat it with peg lipid, and then you're gonna increase the circulation time, but that also means that you're gonna increase the toxicity that it imparts, if it imparts the toxicity. So we need to, it's a balance between the safety and toxicity profile moving forward.
Pegylation of your LNP is one of the major parameters that is going to impact the half-life of your drug product.
The half-life of any oligonucleotide is very, very minimum, since it gets hydrolyzed and it's taken up by nucleases. So lipid nanoparticle encapsulated mRNAs or oligonucleotides will have a better half-life and long circulation profile.
So one of the biggest things that could modify and aid in endosomal escape is going to be the design of the cationic lipid, as well as the chemistry behind that lipid. This will also impact the transfection efficiency as well. Too much of holding on would delay the release, which would make for a longer or suboptimal transfection efficiency, but too little and it may be unstable for the particle.
So the processes become a little bit more complex just because you have with each different LNP, you're going to have differences in processes that you're going to have to make. So altogether, I would say the three main pieces of the downstream process are vital to the LNPs.
People do say that mixing comprises of the most critical step, but to maintain the stability of your particle after mixing, purification, filtration are all very, very critical unit operations that everybody should be thinking about. Yeah.
So the downstream operations and processes in LNP design and manufacturing is absolutely critical. Once the particles are made, it needs to be diluted or needs to reach a specific pH or conductivity in order for the particles to remain stable.
So all of those downstream unit operations are important. They're important to control, they're important to not impact certain critical quality attributes of the product. I'll add a plug to say that there are quality attributes of the product that I think in general we don't consider very often, but are also equally critical. Aspects like sterility, aspects like endotoxin, aspects of container closure integrity, of lot homogeneity, and these are all critical quality attributes of the overall drug product when it's made.
LNPs are probably the most complicated drug products that humans have ever tried to manufacture, and steps that you would hope would be inconsequential almost never are. So controlling those processes the best we can is critical to creating a drug product that scales from the bench to the bedside, as they say.
Your LNPs have to be stable across these unit operations. If it's not, it's very hard to scale it up and make it into a clinically viable product. Yes, research to translation is the key is stability, colloidal stability of the LNP. Yeah.
Just changing the lipid ratios by a teeny tiny amount, or the molar ratios, does affect the encapsulations and the way it's formulated. So it's all on the formulation scales. So yeah.
So what I knew here about the scale-up processes, which I would have not known before, there's something very vital information I got from this conference about the scale-up processes and what could go wrong in the process.
LNP is a very small and tight-knit specialty. It was a specialty occupation, but now everyone because of the success of the product on a global scale, it's growing, and it's pretty exciting to see it becoming more center stage.